Well, thank you again for coming to this session. I'm going to share with you some work I've been doing using the DriverMap platform in transcriptome profiling in a drug screening effort. Once again, I want to repeat, plenty of seats up front. I'm not really planning on calling on anyone, but if I do, it'll be people in the back. So. All right, so in drug screening, just to give you a refresher, there are sort of three main phases. There's usually a screen of some sort, either enzymatic, chemical, or some biological function or phenotypic change. I'll be showing you the phenotype of reduced proliferation of tumor cells. From testing a whole bank or library of compounds, you get some leads that seem to work at that phase. And usually the next one or two somewhat related phases are to figure out, well, what is the target, molecular target of this lead, and what might be its mechanism of action. And that's what we'll be focusing on in our example today. And to be more specific, we have been testing compounds, for example, to limit the proliferation of tumor cell lines. And in this graph, we see the proliferation of the A375 melanoma cell line. And the far right shows the vehicle DMSO, that's sort of the control. And we have 5 compounds with variable activity. All of them do something good, reduce the proliferation and viability of this tumor cell line, but they differ. And ultimately, we'd like to know more about what are their targets, what are their mechanisms of actions, these, as of now, unknown compounds. And just to give you some context, this is part of a larger effort where one hopes, or I hope, and there's a lot of interest in this field, to use transcriptional signatures to improve the ability to predict the efficacy as well as the specificity and the mechanism of action of novel cancer drugs in the cancer field, but also in other fields, immunomodulators, et cetera. And ultimately based on this in vitro transcriptional profile. The specific strategy that I'll share with you is to do transcriptional profiling using the DriverMap platform that you heard about on cancer cell lines after in vitro therapy. And I'll share with you some of the progress we've made in figuring out some common and unique signatures and some data at the pathway level as well. Now you heard about the DriverMap platform. If you were eating lunch, here's a 2-slide reminder, a refresher of the DriverMap platform. And they're almost identical slides, but here are the highlights if you ask me. And they're mostly in the text part. You can see the workflow in the schematic, but the key points are that it's an assay that quantitates up to about 19,000 genes in a very simple single tube assay. Requires relatively small amounts, very compatible with small biopsies, whole blood, and other work that I'm doing, for example, and in the kind of small numbers of cells that are in these in vitro screening assays that I'll share with you. And there are a number of other features that are very appealing. It has built-in calibration standards. Alex didn't go into this in great detail, but he has intentionally reduced the signal from the very abundant ribosomal genes, which often swamp your signal in standard RNA-Seq, so there's less of that issue. You get to pay more attention to the genes you're interested in. And there's also a similar turning down of the signal from hemoglobin, so you can go in whole blood without any hemoglobin depletion needs. Okay, and he mentioned a number of the applications. I just put in bold the one that I'll be sharing with you, which is you can use this technology to profile drug responses and learn something about what's going on. There's plenty of other potential. My favorite one, to tell you the truth, is number 2, which is the idea that you can use this human-based platform to study human tissues in PDX or other xenograft models without having to worry about the mouse background. All right, so here's the experimental protocol I used for these studies. We took a number of cancer cell lines. For example, A375 is a standard melanoma cell line. A549 is derived from a lung adenocarcinoma. They were plated at a relatively low number per well, 5,000. They were treated with vehicle or a panel of compounds at 0.1 to 10 micromolar, standard kind of ranges for this kind of operation. And we had replicate plates. one set of plates were harvested at 6 or 24 hours, for example, for DriverMap profiling of the transcriptome. Another plate was let run for 48 to 72 hours to look at the effects on cell viability and proliferation. For the transcriptome data, here's a summary of the analysis workflow that I used. From the DriverMap platform, what you get is basically a spreadsheet of genes in one column and the counts per sample, not unlike the final product of an RNA-Seq analysis. I normalized the data. I chose to filter out counts below 20. These are sort of the very low or unclear significance genes. Transformed it to log2, and this allowed me to use a third-party software, ClueCore, which I find to be terrific for analysis of gene expression data. That software was used to identify differentially expressed genes, and then also principal component analysis as well as gene set enrichment analysis can be done in that software. The gene lists that came from the differential gene expression analysis were used to query databases that are instructive for analyzing drug signatures, specifically the Lynx 1000 database. I'll say a little bit more about it in a moment for those of you who aren't familiar with it. Commercial platforms like Ingenuity IPA to analyze mechanistic signals and pathways, and an open source pathway analysis tool developed by Avi Mayan at Mount Sinai called Enricher, which is terrific. All right, so here's the data to be analyzed in our example case. We have a set of compounds that seem to have some activity. Can we learn something more about how they're working? Having done the transcriptome profiling, we basically observed roughly between 1,500 to 2,500 differentially expressed genes in each drug versus control at twofold change or greater at an FDR of 0.01, which is, so there's a lot going on in these cells. We were able to cluster the compounds using PCA, and the main message from this kind of analysis is that we have very good separation from our control, the DMSO in green, of all the compounds. They're different from each other, and all the replicates, there were 4 replicates, each clustering together almost like one of these molecule models, are very closely and tight, showing that the technical replication is very satisfactory. You can also analyze these differences by heat maps and looking at the clustering of gene expression. On the left panel, you see 3 of the compounds versus DMSO, and you can see there's clearly differences in expression from the control and from between each other. There's some similarities and differences, and the same applies on the right panel. I broke them up into 2 because it was just too overwhelming to have them all in one heat map. All right, so since we're after trying to, in this example, figure out what can we learn about these compounds, what might they be doing in terms of mechanism of action, I turned to the Lynx 1000 database. And how many here are, I promised I wouldn't call on anybody in specific, but broadly, anybody here familiar with Lynx 1000 or the connectivity map tool? It's an effort at the Broad Institute where they have a huge effort to take perturbagens, approximately 10 to 20,000 compounds tested in mostly now 9 cell lines, but up to 20 cell lines in the database at 2 or 3 concentrations and 2 or 3 times. And they do transcriptional profiling on all of those units. It's a huge database, and they've recently added about 20,000 knockdowns and about 20,000 overexpression data points as well. And it's a fantastic resource to look at the effect of drugs on transcriptional profiles, at least in vitro. All right, so what they have developed is a tool. You can come in with your own data set, as I have, with things that are up or down for each drug. Enter the 150 genes up, 150 down, that's their limit, the top most differentially expressed genes. And they will match your signature to what they have learned for their extensive analysis. And the good news is they in fact have the A375 as one of their cell lines that they've analyzed. So by doing that, we found that our so-called unknowns, V, P, B, and A, were mapped to the top mechanism of action from the Lynx was V looks like a RAF inhibitor, and in the far right column, in the parentheses are the ranking of number of drugs with that type of activity that allowed the database to identify this mechanism of action. P and B both look like HDAC inhibitors. A was a heat shock protein 90 inhibitor. And then we had one compound that had a number of different mechanisms of action, as opposed to really emphasizing one. Now, I've been leading you astray a bit because, in fact, V, P, B, A, and Y were not really unknowns. They were drugs we had chosen, and we knew what they were, but we were testing the system. But for argument's sake, this is as if we showed up with unknowns and we asked the system, can we identify how they're working? V is vemurafenib, which is a RAF inhibitor. That was the top category of mechanism of action identified with this query through the Lynx 1000. P and B are panibatat and belinostat, both of which are HDAC inhibitors. The top mechanism of action identified in this exercise was, in fact, an HDAC inhibitor. And you can see their ranking in the drugs is not panibatat and belinostat, for example, came in as number 3 and number 6 in similarity. But that's good enough to get you the mechanism of action, given the vagaries, the slight variability in gene expression that we all know about. A is AUY922, an HSP90 inhibitor. That mechanism of action was also identified. And the curious case of Y, which is YM155, initially described as an inhibitor of a molecule called survivin, but more recently recognized as having multiple targets. It's not a single action drug, and those targets include BCL inhibition, HIF modulation, and AKT inhibitor, and all of those were actually identified in this exercise. So the conclusion from this was that this was, if you show up with a list of unknown compounds, or compounds you have really no idea about the mechanism of action, this is encouraging that you could, at least as a proof of principle, you could take this approach and at least get a clue to then pursue further action about verifying that this is what you're working with. And this all derived from relatively easy to perform 96-well plate using the DriverMap transcriptome assay that we're talking about. And here they are with their identities revealed in that original PCA. Now, let's talk a little bit more about other things you can do to learn more about unknowns. Let's take the case of the drug vemurafenib, which is the RAF inhibitor that was one of our panel that we talked about. This is a very interesting drug developed for melanoma recently. It is a specific inhibitor of a mutated BRAF gene, the BRAF V600E mutation, which constitutively activates the signaling pathway outlined on the slide, leading to excessive cell growth and proliferation of the melanoma. And it's part of a great excitement clinically in the field of targeted therapy. Oh my goodness. Okay, that one I didn't see. Well, the image with just the skull cut off above the credits is a picture of before and after therapy. In 15 days, this individual, most of their melanoma resolved, metastatic lesions with vemurafenib. Hence the interest in this kind of drug discovery. And I apologize for the problem with the image. Now, another approach to learning more about what your drug is doing is to take your differentially expressed gene, and there are commercial platforms like Ingenuity, and they have one branch of their toolkit called causal analysis. And when we interrogated our gene list from these drugs, the highest negatively activated or inhibited molecule with treatment with vemurafenib was RAF kinase, the kinase that it inhibits. So that was very satisfying. In addition, the next three drugs, the RAF kinase in this melanoma that has that mutant molecule was deemed activated or indeterminate, so it was a reasonable control. It's not telling you everything is inhibited. And finally, AUY922 also gave us an inhibited signal, But in fact, AUI-922 is an HSP90 inhibitor, which is known to inhibit RAF kinase. So that was actually a good outcome, or an appropriate call by this analysis. Now, I also want to tell you that we've done similar analyses with other cell lines. For example, with A549, the lung adenocarcinoma cell line. Here's a similar initial screen of a few compounds for effects on cell viability. And you can see the far right DMSO is the control. We have 2 drugs that don't do much, and 2 drugs that do a very good job of knocking back this cancer cell line in vitro. And when we did DriverMap transcriptome profiling on these same cells with the same agents, you see something I think is quite interesting and very encouraging for this approach. The clustering is the vehicle, DMSO, and the 2 drugs that do nothing essentially are all on top of each other. They're really identical, which is compatible with the functional outcome, and the 2 other agents are separate from those and from each other. And we have similar data to what I just shared with you with the melanoma example, but I'll just stop here for this. Now, in this spirit of taking DriverMap transcriptome profiling and then playing with the subsequent data using available bioinformatic tools, one can keep going. And how many of you here do some bioinformatic analysis of RNA sequencing data or genomic data? At least one person? At least 2, 3, okay. The rest of you are too shy to admit it, but you probably do it too. Okay. So, and this is a slide for Paul because I'm sure he's worried that I'll now show 10 to 30 slides of further bioinformatic analysis, which you can do with almost any set of genes outcomes. Here's fearless Dr. Alice going after more and more analyses, but I'll only show you 2 before we're going down the rabbit hole. You can take the same kind of data and query pathway-based tools like Enricher or Inside Ingenuity IPA to compare pathways that are common or drug-specific. Here's a panel of agents tested with the melanoma cell line, and these are down-regulated. But if we look at, and you can't see the names of the pathways, but the message is that while they share some pathways, each drug has some distinct individual effects that you can use to discriminate their biology, or you could use to discriminate unknowns. And the final slide along this same kind of line of thinking is, for example, there are 3 well-known RAF inhibitors besides the vemurafenib that I've discussed with you already. And on the left, as I've shown you before, this is upregulation, but they share some commonly upregulated pathways, but they actually have distinct signatures despite all being RAF kinase inhibitors. And this is common for drugs. They all have their own little world of unexpected effects. You can also use this kind of data to deduce the transcription factors behind the transcriptome changes. And these drugs share a number of commonly activated transcription factors and some unique transcription factors. And the final possible utility of this is this kind of transcription factor analysis. You can go backwards and say, well, what are the genes that were upregulated that led to the calling of this transcription factor set? And those are bracketed out here. And so, for example, for that first drug, dabrafenib, this list of genes could be a pool of potential biomarkers for response to that drug and not another. And so you could work this kind of information even further to get to a clinically useful outcome. All right, so let me summarize. On the methodology front, you've heard a lot about DriverMap as a tool. It's similar to RNA-Seq, but it's focused on the protein coding genes. It's reasonably described as faster, cheaper, and easier. There's a whole range of applications that you've heard about and hopefully that you're thinking about for your own work right now. The results I've shared with you are one example of this kind of platform in use, looking at drug response transcriptomes in vitro. And this allows you to generate signatures at the gene level, differentially expressed genes, which you can then use to characterize mechanism of action. little more speculative is you can use this kind of signature to generate pathway information and transcription factor identification for biomarker discovery. That's in the future, but I think the overall approach has a lot of promise for those outcomes and for predictive signature development. And I'll just finish by thanking two folks at Cellecta, Alex Chenchik and Gus Frangu, for their help with this work that I've told you about. Thank you. Glad to take any questions.